All right, so in our last lesson, we looked at people who studied DNA. Now we're going to look at some of the things that they discovered, not all of them, but some of them. We're going to take a look at the structure of DNA, which you've heard lots about already, and this mysterious other RNA that I briefly mentioned before. So the first thing that you need to know about are these things called nucleotides. Nucleotides are what we could call the subunits of DNA. They're the things that make it up. So you all had to take Bio30 to be in this class, and so I know you would have heard about subunits of things like proteins. The subunit of a protein are amino acids. The subunit of carbohydrates are monosaccharides, simple sugars. The subunits of lipids are glycerol and fatty acids. This is the same idea. These are the pieces that make up DNA. Now the picture that's there is the same one that you have if you printed things out, represents the 4 different nucleotides that make up DNA. They have some things in common. Each of them has a 5-carbon sugar. If you look at each of these pictures, you can see like a pentagon shape here. That is the 5-carbon sugar. It's in a pentagon shape because there are 5 carbons. Now, some of you may have taken chem already, and some of you maybe didn't take chem or aren't taking chem, or you're taking it right now. When we talk about organic molecules, ones that are based with carbon, we will frequently not draw all of the atoms that make up the molecule, but every line represents a carbon here. I'm only telling you this because some of you like to know these things. The chemistry of these molecules is not the biggest focus here. The 5-carbon sugar is deoxyribose. It's the D in DNA. So that's in common. All 4 of those nucleotides would have that same sugar as part of their structure. They all also have a phosphate group. So if you look, there's a little line and a phosphate group attached. So far, those 2 things are identical in each of the subunits. The next thing on the list is what distinguishes them, what makes each subunit different from the next. It's the nitrogen base. So if you look on each of these, there's this piece at the top that I'm just going to circle. That's the nitrogen base. Now, even though each DNA nucleotide has a nitrogen base, you'll notice there's a different letter in each of the pictures. They don't all have the same nitrogen base. Now, when we're talking about DNA, which we are right now, there are 4 different bases that we could use. Adenine, which is the A here. Guanine, which is the G. Cytosine, which is the C. And thymine, which is the T. So very frequently when we talk about DNA, we will use just the letter A, T, C, or G to tell you which nucleotide we're talking about. There's a 5th nitrogen base that wouldn't show up in DNA. It's called uracil. And we'll see a couple slides later that when we talk about this other molecule, RNA, instead of having thymine, we'll have uracil. So if someone were to ask you what are the parts of a DNA nucleotide, those 3 things that I colored blue and green and red, those are the 3 parts. Does anyone want to ask anything about that so far? Okay. You don't have to memorize these names. You might recall that I gave you not last week, 2 weeks ago, this data sheet that has a bunch of tables of stuff on it. And we looked at the back of it There's a little chart, and underneath it, it lists the nitrogen bases that make up DNA. It tells you what their full name is. So if you forget what's the A in DNA, it tells you on your data sheet. It also tells you what classification they have. And so I'm just going to mention that right now. If you look at my pictures, and these are just sketches of how it might look if we were going to draw this, you might notice that adenine and guanine kind of look the same. They have the same basic structure, and that thymine and cytosine sort of look the same. Adenine and guanine have 2 pieces in their nitrogen base, and thymine and cytosine just have 1. Adenine and guanine, and it says this on your data sheet in case you ever need to look it up, are what are called purines. And thymine and cytosine are pyrimidines. And I'm telling you that because when we look at the next slide, it will be kind of important that there were 2 different types of nucleotides. Now, one of the people that we mentioned at our table a couple weeks ago was this guy named Chargaff. And he discovered something about DNA. People thought originally that there should be 25% of each of those in a person's DNA. And he realized that that wasn't the case. There wasn't equal quantities of each of them. He also realized that it was not constant or repeated in its sequence. So originally people thought, well, it must just go in this repetitive order, A, T, C, G. I would have the same amount of each nucleotide. But Chargaff is one of the people that realized, no, that's not the case. Chargaff's rule includes what's written in that bullet there, that it's not constant, not repeated, but also the point down below. Adenine is always equal to thymine, guanine to cytosine. So I wrote down the other day when we were making our list of scientists that A was equal to T and C was equal to G. So I'm just going to show you the little very bit of basic math that would go along with this. If I told you that in a person's DNA that we analyzed, 20% of their nucleotides were A, adenine, and then I asked you, what percent of thymine nucleotides should I find? What would you tell me? I hope you would say 20%, because these have to be equal. So Chargaff is saying, if we look at someone's DNA and we figure out what proportion is A, the same exact proportion should be T. Now, how much C and G are there? And you want to tell us how you figured out that it was 30%? So that exact thought process, you have to have 100% total. I already have 20% A and another 20% T, so that makes 40%. Out of the whole 100%, that leaves me 60%. So I divide 60% in half. 30% must be C, and 30% must be G. That is an example of an actual number that could be the answer in a numeric response question. So every once in a while, something like this would show up. We'll tell you what percent we have of one of the nucleotides and then ask you about another one. Now, does anyone want to ask anything about a nucleotide or about Chargaff's rule? So next, I purposefully put 2 different pictures that don't look the same of a DNA molecule to show you that different artists have different ways of representing things. The picture on the top is a little more artistic freedom, and the picture on the bottom is a little more closer to the correct chemical structure. If you ever have the urge to draw a segment of DNA, it doesn't matter to me which angle you take. If you want to draw it more like the artistic person, or if you want to draw it more like the chemistry person, they're both showing me the same things. Now, here are some of the things we know about DNA. First, it's a double helix. Double means 2, Helix is like a twisty structure. Think helicopter, how it has the spinning propeller on the front. So a helix is telling us sort of that idea. Since DNA is a double helix, it's like two strands that are twisted. A lot of the time people will say it looks like a twisted ladder with pieces on the outside and then one look like rungs going up the middle. Now, how do we know this about DNA? Mostly because of Rosalind Franklin. She's the one who took X-ray photos. And then Watson and Crick, the guys who made the model based on the findings from all of her research. So here's what we know. DNA is a double helix, and it has sugar and phosphate on the outside. So if you look at these two pictures, on the picture on the top, I have what looks like stripy lines on the outside. Every second piece is the sugar piece. On the picture on the bottom, I have these pentagon shapes, kind of like the nucleotide pictures I showed you before. So here's the sugar. There's also phosphates on the outside. So in the picture on the top, that's a little more artistic than chemically correct. It just looks like this alternating band of two different pieces. The fact that I colored them blue and green is not relevant to life. That is just the colors of smartboard ink that I have. But the fact that they alternate and that they're on the outside is important. On this little picture, the phosphate already is green. And you can see them as these little circles here. So if the sugar and phosphates are on the outside, the nitrogen bases are on the inside. So the nitrogen bases, like we just listed, are the A, the T, the C, the G. On the picture on the top, each shape represents a different nitrogen base. So for example, the pointy one is thymine. The one that's like the opposite or complementary shape to it is adenine. Cytosine is this one, and guanine is that one. So in that picture, we're showing that they have like an opposite shape to each other. The picture on the bottom is just showing them as molecules attached by a little dotted line. Now, the other thing we know from Rosalind Franklin's photos is that the distance between the phosphate and sugars is constant. So it's not a molecule that gets really wide, then really thin, then really wide, then really thin, but it's a constant width as we go through. Now that's interesting because the size of the bases is not all the same. The size of the bases is different. So on the previous slide, I showed you that adenine and guanine were kind of bigger in their structure, and thymine and cytosine were a bit smaller. But we just said that there was always a constant space between the two sides of my DNA molecule. So that tells me that those bases must be paired up in a certain way. They form what's called a complementary pair. The picture on the top shows it better. Even though it's less chemically correct, it shows this idea of a complementary pair. A and T, adenine and thymine, are complementary. And in the picture on the top, you can see their shapes are made to look like they're exactly the opposite, like they would fit together like two pieces in a puzzle. What's important is that one of them is a purine and one of them is a pyrimidine. One of them is a little bit bigger and one of them is a little bit smaller. The same thing is true for C and G. One of them's a bit bigger, one of them's a bit smaller. So when they fit together, we always end up having the same space between the two sides of our DNA molecule. The thing that attaches them are hydrogen bonds. Now I'm sure some of you took chem and maybe someone remembers what a hydrogen bond is. Yeah, Martin, you remember? Very nice description. So it's when hydrogen is attached to something that's super electronegative, like oxygen. And that's what it is in this case, it's oxygen. It could be nitrogen or fluorine as well, but something that's really electronegative. And the really electronegative atom pulls the electrons with such force that the hydrogen is left basically bare, stripped. All you have is its nucleus that has a proton in it, but no electrons guarding or shielding the nucleus. Then hydrogen can form these really strong intermolecular forces. So I'm just going to add here that this is an intermolecular force. Even though it's called a hydrogen bond, it's not a full chemical bond, not like an ionic bond or a covalent bond. And so hydrogen bonds are usually drawn with some sort of broken or dotted line to indicate that it's an attraction between the two sides. If you look at the picture on the top right here, those are the hydrogen bonds, those little dots. And on the other picture, the dotted line in between the two sides, those are the hydrogen bonds. So when we say that DNA is complementary, it's because we have a purine on one side, pyrimidine on the other side, and they're attracted to each other, held together by this intermolecular force of a hydrogen bond. The other thing that's true is that the two strands are what we call anti-parallel. Now the top picture does not do a good job of showing this. The bottom picture does a better job. To explain what anti-parallel means, you need to know a little bit about chemistry. So I'm just going to briefly explain this, and if you would like to ask more, I would be happy to explain it to you after. In chemistry, when we're dealing with carbon-based compounds, we have to number the carbons so that we know how to name the compound. So on these little structures on the side here that have sugars in them, each corner of the pentagon is where there would be a carbon, and we would number each of those carbons. And there are lots of rules for how you number the carbons, and that is not the point of this lesson. That's just where it's coming from. When we say the two strands are anti-parallel, it's because their backbones, so the part that's made up of sugar and phosphate, have opposite directions, 5' versus 3'. So what we're saying is, essentially, the way the sugar and phosphate are oriented is backwards. If you look at this picture, it does a good job of showing it. On this side, see how the sugar is like pointed upwards, and on the other side, it's pointed downwards? That's how we know which way things are going. I'm just going to pause here. Okay, so this next slide is about RNA. The other day, like 11 days ago when we did that lesson, I told you about Phoebus. He isolated DNA and RNA, and we said that DNA was deoxyribonucleic acid, and RNA was ribonucleic acid. RNA is kind of like DNA. They're pretty similar in a lot of respects, but there are some things that are different between them. Now the picture that I'm showing you is a specific kind of RNA here, just so that you're clear, because there's more than one kind. The one I'm showing you is mRNA, and the next slide will identify some of the types of RNA. If you look at the image, though, and you think about what the picture of DNA on the last slide looked like, there are three key things that are different. The first thing is what the sugar is. So when I look at this strand and I look at the backbone, it still goes sugar, phosphate in an alternating pattern. That's the same. DNA had that as well. Sugar and phosphate on the outside. The thing that's different here is that the actual sugar isn't the same. It's ribose, not deoxyribose. The second thing that you might notice has to do with the bases. There's no thymine in RNA. If you remember what the pieces looked like before, on the picture of DNA, this is what thymine looked like. And so on this one, kind of looks like thymine, like it still has the pointy end, but the shape has been changed so that you can see that it's different. This, remember, is just an artist's representation so that you can see that it's distinguishable. So when I look at the bases that RNA has, RNA would be made up of A, U, C, and G, whereas DNA was made up of A, T, C, and G. Because we've taken away thymine and replaced it with uracil, A and U are complementary in RNA. Now the third thing that might make you wonder why I said what was complementary to what is that RNA is made up only of a single strand. DNA was two strands. That's why we called it a double helix. It had two sides. RNA looks just like this, where it's just one strand. Sugars and phosphates alternating, and then those nitrogen bases attached to them. So you need to know how to distinguish DNA from RNA. It is the topic of lots of questions if you look at old diploma questions. You need to understand, maybe by looking at a picture, oh, the one with two strands is DNA, the one with only one strand is RNA. You need to understand, especially if we're representing which bases there are, if there's a U, you're looking at RNA. If there's a T, you're looking at DNA. And if the sugar is named, you can use that to distinguish them. Visually, it's hard to distinguish the sugars. Their chemical structure is really not that visually different, and a lot of the time they aren't drawn to look different in terms of shape. But it might come up in a question where words are written. So sometimes I will ask a question where I have a big list of characteristics, and I'll ask you either which characteristics do they share? So which do they both have? For example, they both have a sugar. It's not the same sugar, but they both have a sugar. They both have A and C and G. It's just the T and the U that are different. I might ask you which characteristics are unique to RNA or are unique to DNA. And you'll see that one of the questions you have to answer is where you make a little Venn diagram to compare them so that you can practice that. Does anyone want to ask anything about RNA's structure here before we go to the next one? So there are multiple types of RNA. The three that I'm showing you are the three big types here, but there are actually more types if you study genetics in any sort of real big detail. The first type is the one that we just looked at. mRNA, where the M stands for messenger. Kind of looks like a ribbon or tape. Now, messenger RNA, like its name suggests, is a messenger. Earlier today, we talked about why people maybe thought DNA was not the molecule of heredity, and it's because it doesn't go anywhere. But messenger RNA is this cool little molecule that can be made from DNA and that can carry the code for making proteins to other parts of the cell. Now these two words, transcription and translation, they are not me just trying to be excessively fancy. They're the actual words that we're going to use when we talk about this in a couple of lessons. But we're just trying to get across that the point of all of this, the point of DNA, is that it has instructions for making proteins. DNA doesn't leave the nucleus, but this messenger RNA can. There's another kind of RNA. It's ribosomal. so rRNA. They're the main constituents of a ribosome. So you're going to look in your vault in your brain and say, ribosome. That was that thing that we learned about in Science 10. It looked like a little dot attached to the rough endoplasmic reticulum. So you might have drawn a ribosome just as a dot before. If you zoomed in on that dot, this is what you would see. Two little pieces that fit together. So it's not really just a little circle. It's actually these two sections. And if you see this sort of ribbon in each of the sections, there's the RNA. That's what we're trying to represent there. And we're mentioning this one because ribosomes are actually pretty gosh darn important. For how big they are, or rather their lack of bigness, they have a really, really, really important job in terms of making proteins, which, like we said, was the point of this. The last type of RNA that I'll mention is transfer RNA, so transfer or tRNA. They kind of look like a clover leaf if you look at the top. So see here, it kind of looks like a clover leaf at the top. It's really just one strand, but it's all folded in on itself, so it sort of looks like a clover leaf. They're important because they each carry an amino acid. So that little ball at the top, that's the amino acid. And if you remember, Bio20, amino acids, those are the building blocks of proteins. And we said that making a protein is going to be the point of this. So transfer RNA will be really important when we get to building the actual protein. They're going to carry the amino acid so that it can be integrated into a protein. So for now, I just wanted you to see what the RNA looks like. We will talk a lot more, especially about the M and the tRNA, when we get into the mechanics of how a protein is being made. But for now, we're just looking at structure, really. The last thing I want to talk about when we're talking about DNA structure is the sequence. By sequence, we mean order of nucleotides. Way back when people didn't think DNA was the molecule of heredity, we thought that maybe it just went in the same repeating pattern. But what we know now is that the order of nucleotides, that's the thing that makes things happen. Different orders of nucleotides make different end results in the cell. So we've used the word gene before. We used it when we talked about chromosomes. Chromosomes have genes. A gene is this functional piece of DNA. And I say functional, and I maybe put a star here, because that's the key thing. A gene has some functional thing that it's going to accomplish. A gene is this section of DNA. Remember, your chromosomes are just a bunch of DNA, so it's a section of one of your chromosomes that will direct the production of one or more polypeptides, which is a protein. And that's what makes cells different. Different cells make different proteins, so they look different. They have different properties. Your cells have different genes than somebody else's cells, so the protein that makes the color of your hair or the color of your skin is different. Now, a gene is just a little section, right? Each of your chromosomes could have thousands of genes on them. Your genome is more than that. It's the sum of all the DNA, everything. That would include a gene and non-coding sections. So if we were to look at a chromosome that a person has in one of their cells, there will be spaces on the chromosome where there is DNA, but it's not coding for anything. It's not directing the cell to make a protein. That's what a non-coding section would be. Genes are not evenly spaced. So sometimes on a chromosome you might have 5 genes in a row and then a big chunk of DNA where it's not telling the cell to make anything. And there's no specific pattern or spacing about genes, about which sections of DNA code for a protein and which ones don't. There is a ton, and when I say a ton, I am not exaggerating, a ton of research. Humans and all the other species, because we're just one sort of mildly important species, but there are tons of others. But there is so much research into DNA sequencing. So the process of determining the exact nucleotide sequence of a gene, an entire chromosome, or an entire genome. Maybe you've heard of the Human Genome Project, sequencing all of the chromosomes and all of the genes that humans have. It's a big task. If you were thinking about your de facto, for some of you it's coming up sooner than others, and you were like, I don't know what to look up, if you were interested in genetic engineering or genome sequencing, you could find research that is current because this is ongoing. It's not something that happened once and is over, but rather ongoing. People are constantly researching new things, finding new genes here.